Prion diseases affect wild animals, farmed animals and humans. My name is Benestad and I'm working at the Norwegian Veterinary Institute on prion diseases in animals. For the WiliMan-ID project, I'm focusing on chronic wasting disease. Chronic wasting disease, in short CWD, is a highly contagious prion disease. It is only detected in cervids. As of today, it is unknown whether other mammals or even humans could be infected. In 2016, the first case in Europe has been discovered in Norway. So we must study whether there is a risk of spread across Europe and if it is a danger for humans. CWD is not caused by a bacteria or a virus or a parasite. It works differently. Unlike the 4 viral diseases that are investigated in WiliMan-ID, chronic wasting disease is a prion disease. Anyways, it shares similarities when it comes to strains, mutation and factors that can influence their aggressivity and their ability to contaminate other hosts. This is a normal prion protein, as there are many in the brain of all mammals. Very seldom it happens that this protein can misfold. And then there is a kind of chain reaction. Some protein that comes in contact with it also adopt the same misfolding. They are converted into prions, they become resistant to body enzymes. And progressively these prions accumulate and form aggregates. And these aggregates cause severe damages in the host brain. It ends with the death of the individuals. CWD was first detected in 1967 in Colorado in the USA. 30 years later, cases were detected in Canada. The disease came with a deer from a zoo in Colorado that was brought there. 4 years later, same scenario in South Korea, this time from a zoo in Canada. After 40 years, we see new hotspots: Illinois, New Mexico, Alberta province. Within 8 years, CWD persistently spread more and more. Nearly 60 years later, CWD is present in 34 American states and 5 Canadian provinces. Here is the first European case, detected in 2016 in this Norwegian wild reindeer. Ever since, we have also identified the disease in moose and red deer. Neighboring Sweden and Finland have also detected CWD in moose. From North America, we know that CWD is a contagious disease, spreading from one deer to another deer. But how? The contamination can occur between living animals through direct contact, but it can also happen indirectly through the environment. Sick deer can contaminate soil and plants with their urine and feces containing prions. Also, the carcasses of dead infected animals stay contagious. Prions are extra resistant to most decontamination procedures and can still be infectious after years in the environment. This, of course, represents a challenge to eradication measures. It needs to be said that all the data we have about the contagiousness are based on CWD from North America, and we found out that in Europe we have different strains of the disease. So we are currently investigating how it is with the Norwegian CWD strains. The disease is difficult to spot because of many reasons. It has a long incubation period where the deer seem to be healthy. In contrast to this, the period where signs of the disease are visible is short. Moreover, it affects mostly wildlife animals living in remote areas that no one can observe. The signs of the disease seen at terminal stages are mostly loss of weight and behavioral changes, among them loss of human fear. The disease takes time, but is always deadly. It cannot be cured or prevented by vaccine. In WiliMan-ID, we have several focus points. 1. To optimize tools to detect small amounts of prion in the urine and feces of the infected animals. 2. To identify which gene can be susceptible to infection and which can be resistant. And 3. To build models for CWD spread in reindeer population to better understand the disease dynamic. Fortunately, we have already diagnosis tests in place used in surveillance program in Europe. In the frame of the WiliMan-ID project, we use newer methods that are much more sensitive and exclusively used in research. Studying which factor can slow down the spread of the disease, as well as enhance the efficiency of detecting contamination, will hopefully help us to sustain healthy wildlife as well as livestock in the future.